Okay, so under the short video series, this is the first topic, that is bacterial promoters. This is the usual disclaimer that this PowerPoint presentation is meant for quick revision and educational purpose only. It should not be used for any other purpose, copied or posted on any site without the permission of the author. And some of the pictures in the presentation may have been taken from public domain websites or popular books. So they should not be, they have been adequately acknowledged. Okay, so what exactly is promoter? Promoter is a short stretch of DNA, approximately 60 base pair in length, where RNA polymerase binds and initiates transcription. Where is it located? It is located upstream the structural genes. So let us understand what is meant by upstream. So let's say this is the start site. This is ground zero. On this side, this is called as downstream. And it is in this area that structural genes are located, the genes which are to be transcribed. So this region is called as downstream. Okay, and it is also designated as +1, +2, +3 like this on this side. And this area on this side is called as upstream. Okay, and it is designated as -1, -2, -3, -4 like this. So promoters are usually located upstream the structural genes under their control. So usually promoters will be located here upstream. So what are the characteristics of promoter? Bacterial promoters or prokaryotic promoters, as you can call them as, they're characterized by presence of two consensus sequences. What is meant by consensus sequences? That means when scientists analyzed large number of RNA polymerase binding sites in bacterial genes, they found out that the sequence at these locations, -10 and -35, minus indicates upstream. Upstream the start site, this is the start site. And the sequences were same, identical, and therefore they are called as consensus sequences. So what are the consensus sequences? At -10 you have TA, TA, AT. Whereas at -35, centered at -35, you have the sequences T, T, G, A, C, A. So majority of the promoters, bacterial promoters, they will have these two consensus sequences present, centered at -10 and -35. But there are certain promoters, which govern different sets of genes. They have different consensus sequences. The consensus sequences in those promoters are different from those shown over here. Okay, and as a result of that, they are identified by different combination of core enzyme and sigma factor. So we'll come to this in the next slide. So for identifying these promoters, you will require a different combination of core enzyme, RNA polymerase, and the sigma factor. Okay, so how is it identified? How is it recognized? Sigma factor of RNA holoenzyme, RNA polymerase holoenzyme, it helps in identifying the promoter. Okay, let us understand the structure of RNA polymerase. The RNA polymerase, bacterial RNA polymerase subunit composition is shown over here. It is made up of a sigma factor. Then there are two alpha subunits. There is one beta subunit and there is one beta prime. This is beta prime subunit. Okay, so these four subunits, 2 alpha, beta, and beta prime, they constitute what is called as core enzyme. So this is core enzyme. This is responsible for majority of transcription. Whereas this core enzyme means these four subunits along with sigma, this makes what is called as an holoenzyme. So the role of sigma, this subunit, is to identify the promoter. And once the promoter has been identified, the sigma subunit leaves the complex and the remaining transcription is carried out by this core enzyme. Okay, so how does sigma factor help in identifying the promoter? It does it by reducing the affinity of RNA polymerase for general regions of the DNA by a factor of 10 to the power 4. Okay, how does it do it? It reduces the affinity of RNA polymerase for general regions non-promoter regions of the DNA by a factor of 10 to the power 4. Okay, and the holoenzyme binds, holoenzyme means core enzyme along with sigma factor, it binds to the double-stranded DNA and searches for the promoter region by forming hydrogen bonds with proton donors and proton acceptor groups of DNA. Okay, so it will bind here to the double-stranded DNA. This is RNA polymerase holoenzyme. So it binds here and it searches for the promoter region by forming hydrogen bonds with the exposed groups, proton donor and proton acceptors of the exposed groups of the DNA. And it does it rapidly. How does it do it rapidly? It does it rapidly by sliding along the double-stranded DNA. It slides along the double-stranded DNA and looks for what? Looks for the promoter region. And how it identifies the promoter region? The regions are identified, the promoter region is identified by the presence of those consensus sequences located at -35 and -10. And this recognition of consensus sequences by the RNA polymerase takes place without unwinding the double-stranded DNA. Otherwise, it would have taken a longer time. So it identifies these consensus sequences when they are present in the double-stranded form only. Okay, sigma factor, as I said earlier, leaves the complex once the promoter has been recognized. Once the job is over, it leaves the enzyme. Okay, so there are different promoters. As I said, majority of genes, they will have these promoters characterized by the presence of -10 and -35 consensus sequences as shown over here. TA, TA, AT at -10 and TT, GACA at -35. So they are called as standard promoters. Okay, and these standard promoters are identified by combination of core enzyme. This is core enzyme. You know, there are two alpha subunits, one beta and one beta prime, and there is one sigma factor. It is denoted as sigma 70. 70 indicates what? 70 indicates its molecular mass. So sigma 70, when it combines with this core enzyme, you know, it is capable of identifying all those promoters which have these two consensus sequences. But as I said, you know, you have some promoters where the consensus sequences are different, and these promoters govern different sets of genes. For example, there is a group of heat shock genes, and the promoter is different in the sense that -10 consensus sequence is this, whereas -35 consensus sequence is this. So this promoter, for its identification by RNA polymerase, requires the core enzyme remains the same, but if the sigma factor changes, it becomes sigma 32. 32 indicates what? 32 indicates its molecular mass. So it is different from sigma 70 in terms of its molecular mass. So this combination, core enzyme remains the same, along with this particular sigma factor. When they combine, they will be able to identify the promoters for heat shock genes. Okay. Similarly, we'll take one more example. So there are genes, you know, which help bacteria cope up with the nitrogen starvation situation. So those genes, they have a different kind of promoter. So for example, -10 is consensus sequence is this, and -35 consensus sequence is this. So you have to compare this sequence, these sequences with the standard sequences. So they are different, right? So as a result of that, they will be identified, these promoters, this promoter will be identified by a combination of core enzyme with a sigma which is different, which is called a sigma 54. 54 indicates molecular mass of the protein. So you have three different types of sigma. You know, there may be more, but just to remember these three different types of sigma, they will combine with the same core enzyme, and this combination, different combinations will help RNA polymerase identify different types of promoters based on consensus sequences which are present at -10 and -35 upstream. Okay, this is analogous to what we have a toolbox where, you know, you have one handle and different shafts. Depending upon the screw, type of screw, you know, the handle remains the same, and you can put a different shaft depending upon the type of screw you want to wind or unwind. So this is that kind of a arrangement nature has provided. Okay, then next come is what is referred to as strong promoter and weak promoter. Strong promoter means a promoter from where the frequency of binding of RNA polymerase and initiation of transcription is very high. Those promoters where the frequency of binding of RNA polymerase is very high and frequency of initiation of transcription is very high. Typically every 10 seconds from these promoters, RNA polymerase will bind and will initiate transcription. So these promoters are called as strong promoters. And what are their characteristics? These strong promoters, they're characterized by the presence of standard consensus sequences, okay, without any deviation, without any substitution. So the sequences at -10 and -35, it is a standard promoter, remains same. There is no substitution even of a single base. So such promoters are called as strong promoters. Vis-a-vis weak promoter, where the frequency of binding of RNA polymerase and initiation of transcription is less. Typically more than every 10 minutes, RNA polymerase will be able to bind here and will be able to initiate transcription. In fact, RNA polymerase finds it difficult to recognize these promoters, and many times it will be held by additional factors, and those factors are usually called as transcription factors. So what are the characteristics of weak promoter? Why does it behave in such a manner? Why RNA polymerase finds it difficult to identify? The reason is that in the standard consensus sequences, you have one or more than one substitution. So for example, the one in red is not the normal base present in the standard consensus. If we have A over here in the standard consensus sequence, it has been replaced by T. Similarly, we have a over here in the -35 consensus sequence, we got a G over here. So you have substitution, the promoter becomes weak. You have multiple substitutions, the promoter becomes weaker. Okay, and if you have more than this, it will be still weak. Okay, and such promoters will be very difficult for recognition by RNA polymerase. RNA polymerase by itself may not be able to recognize these promoters and may require additional support, additional help in the form of transcription factors. So this is the concept of strong promoter and weak promoter. So strong promoters will where the frequency of binding of RNA polymerase is high and initiation of transcription takes place almost every 10 seconds. Weak promoters, frequency of binding of RNA polymerase is less and transcription takes place almost every 10 minutes. Thank you.